Now, during the transcriptional process, there are different factors which control the transcription. That can be repression, activation, and so on. Now, in this segment, we will make special attention toward the transcription repression. That what are the different factors which are responsible for transcription repression in the cells. Now again, in prokaryotes, the control of transcription cells that relies heavily on the repressors, which are proteins, and that bind to the DNA and block transcription of a nearby gene. ये जो proteins हैं in prokaryote ये क्या करती हैं? They bind with the DNA and the block the repression of the nearby gene, जो कि जीन है, क्लोजेस्ट जीन है, उसकी ट्रांसक्रिप्शन को रोक देती हैं, वो फिर प्रोटीन नहीं बन सकती. And in eukaryotes, the different mechanism operates, and as we have discussed earlier, that in eukaryotic cell, they possess negative regulatory mechanism to keep the transcription mechanism in harmony. That is, they have got a negative transcription mechanism which keep the transcription mechanisms in harmony. पहले में क्या है प्रोकार्योट में? That the proteins they bind to the nearby DNA, to the DNA to bind, to control or to inhibit the repression to transcription of a nearby gene. Now, there are different switches which are responsible for this. One is the role of acetylation in switching on and off the chromatin. That is, the acetylation, it switches on and off of the chromatin, which is abbreviated as HATS and HDAX. Now, the state of acetylation chromatin is a dynamic property. So what happens? That when a chromatin is acetylated, its property changes. Just as there are no enzymes, histone acetyl transferase, that is HAT, that is to add acetyl groups. These are the enzymes which add the acetyl group. There are also enzymes to remove these acetyl groups. That is, the removal of acetyl group is accomplished by the histone deacetylases, that is HDACS or HDAX. So there are two different enzymes. What is the histone acetyl transferases, that is the HATs, and the second one which have got the reverse mechanism, these are called the HDAX. Now HATs, they are associated with transcriptional activation because they acetyl, and HDACS, they are associated with transcriptional repression, of course. Now HDAX, they are present as the subunits of the larger complexes described as the co-repressor. जो co-repressors हैं वो HDAX में present होते हैं, and they actually what they do, that they cause the targeted gene to be silenced. Targeted gene जो है उसको वो silence कर देते हैं, उसको वो inactivate कर देते हैं rather than के activate करें, ये उसका reverse कर देते हैं. For example, here we can see that the loss of acetyl group and addition of methyl group lead to chromatin inactivation and the gene silencing in the different stages which it takes place. Now, DNA methylation and the transcriptional repression, that is also very important. And DNA methylation of promoters region is generally correlated with gene repression. ये gene repression जो है उसके साथ correlate होती है. And most evidence, they suggest that DNA methylation serves more to maintain a gene in an inactive state rather than a mechanism for initial inactivation. तो ये क्या करता है methylation? ये उस जीन को inactive form में रखता है rather than to make it inactivation. यानी उसे inactivation नहीं करता, उसको inactive form में रखता है. For that. And genomic imprinting and transcriptional repression is also an important mechanism there, and their genes are thought to become imprinted. एक दूसरे को imprint हो जाते हैं as the result of selective DNA methylation of one of the two alleles. एक या दो allele जो हैं, when they are selectively methylated, एक दूसरे को selectively methylated होते हैं, then they are imprinted. एक दूसरे को वो जिस तरह imprint नहीं होता वो बन जाता है, and that is the genomic imprinting and the transcriptional repression. Now, in this process, it involves an acquisition of a closed chromatin state and DNA hypermethylation in one allele of a gene. जीन के एक allele में ये methylation हो जाती है. कब होती है? Early in the male and female germline. मेल और फीमेल के germline की early stages के ऊपर ये methylation हो जाती है उनमें, and this methylation leads to the monoallelic expression. जाहिर है कि जब मिथाइल होता है, जब methylation होती है तो वो एक allele में होती है, उसको inactive करता है और वो inactive रखता है. तो in this case, this keeps it in a monoallelic expression.